Hello, my name is Daimitsshima in Yamaguchi University. Today, we will talk about dynamic change in ripple-like firing activity of CA1 neurons and compared before and after the episodic experience. Hippocampal CA1 neurons are necessary to process episodic memory in various mammalian species such as rats, mice, monkeys, and humans. In the present study, using mature male rats, we started to record multiple unit firing activity of CA1 neurons in well-habituated his home cage. After 15 minutes recording, he experienced one of 4 episodes for 10 minutes. Then we continued the recording for 40 minutes in his home cage. On next day, he experienced the same episode again to check the memory behaviorally. We focused on the ripple-like firing events. Thousands of ripples generated again and again, but they are always different. No same ripple. Spike analysis shows firings of multiple neurons form a single ripple. This is enlarged view. Hundreds of ripples were generated again and again. After recording, we measured 4 parameters from individual ripple. Amplitude, duration, arc length, and the number of firings, peaks. Let me show you short movies. Before the episodic events, the rat mostly shows sporadic firings in his home cage. You may see some ripples, but it's not very clear. Play again. This is after 10 minutes restraint. We recorded the activity in his home cage. Clustered multiple firings form one ripple, and also you can see silent period. Play again. We used 8 to 10 rats for each episode. One circle indicates the feature of one ripple. Although the rats mainly showed plain and simple feature of ripples before the episodic experience, they frequently showed big and loud ripples after these experiences. Because we also plotted peak data simultaneously, actually these are 4-dimensional plot. If you could, please imagine 4-dimensional space. By analyzing the difference of 4-dimensional plotted region, we found experience-dependent and experience-specific change in the ripple features. We used two-way maneuver for overall significance, and we used post-hoc maneuver for specific comparison. As to the experience-dependent change, this and this seem to be very similar, but this is significant. As to the experience-specific change, male and object looks very similar, but post-hoc maneuver showed large difference. Of course, object restraint and object female were also significantly different. Here we summarized our hypothesis of early encoding process. we found that the diversity of ripple features was not only episode dependent, but also episode specific. Moreover, not enough time today. Episode produced synaptic diversity at excitatory or inhibitory synapses, and the diversity was also episode specific. Further, we successfully recorded spontaneous high-frequent firings, superbursts, occurred many times during emotional episodes such as restraint or contact with female. Important thing is the feature of superburst was also episode specific. I would be grateful if you could check our report. Because the recorded ripples were all different, I hypothesized that the ripples encode experienced episode. In the present study, using Python's algorithm, we comprehensively analyze the similarity of ripple pairs. In dynamic time warping method, the program allows to change the time 20% of total. We can obtain minimum Euclid distance of the pair, and also we can get the minimum root of the pair. By analyzing again and again, we can visualize the ripples as a cluster map. As you can see, similar ripples form cluster, and unlike ripples locate in the distance. We analyzed more than 66,000 of ripple pairs. Single red circle indicates the Euclid distance of one pair. Pre means before episodic experience, and also plotted after the experience. After the restraint or contact with female, you can see many high points indicating the episode produced many unlike ripple pairs. After the contact with male, the Euclid distance slightly but significantly increased. In contrast, the distance slightly decreased after the observation of novel object. This is summary. In previous Tomokage's method in 2019, we extracted 4 features from individual ripples, then plotted 4-dimensionally. By using MANOVA and post-hoc MANOVA, we detected episode-dependent and episode-specific change in the 4-dimensional plot. This was really wonderful for us, but present Seo's method in 2020, we comprehensively analyze the similarity of any ripple pairs. By calculating DTW Euclid distance, we clearly discriminated one of 4 episodes. By analyzing the similarity of pairs, we may know his past experience even in unknown rats. Rather than the feature of single ripple, ripple similarity may be important to express past experience. These results suggest that hippocampal CA1 neurons may express a part of past experience by changing the similarity of ripple pairs. In our lab, Dr. Junko Ishikawa recorded multiple unit firing activity from freely moving rats. Dr. Tomokage used MATLAB, and students Sato and Seo used the Python algorithm to analyze the ripple pairs. Because their results are very important, I would express great appreciation to them all. Thank you for viewing our presentation in Japan Physiological meeting.